So today we start talking about microbiology. Let's talk about what we will be learn in the microbiology. So micro is the word. Usually we talking about something very, very small. Small things. So what means small? Tiny, tiny small. We couldn't see with our eyes directly. We must use the microscope. So that means we study something we must be see in the microscope. And the bell, that means living things. We have lot of living things, including in the bell, like plants, like animals, like a human being. That's the living thing. But if we put the micro and the bell together, means that kind of living things is very small. Then we use the logic. We have a lot of logic. Logic just means study. Logic just means study. So that means in this class, we learn something very, very small, but it is living things. It's not dead things. They still have like metabolism. They can grow things. They can reproducing. They can moving. That means living. So that's basically in this class. So the commonly things I believe you are familiar with, like we're talking about bacteria, like we're talking about virus, and sometimes we're talking about fungus. So that's few pretty commonly category in there by their shape and by their like attractors. We separate the small, tiny living things by different categories. So that's basically we will learning in this class. So why we will learning these micro things here? Because these kind of micro things will cause the people illness. So that's why we need to study. We study these micro things and to learn how they cause the people getting sick or ill, then they can prevent it. So that's why we need to learn. So that's the first part we're talking about in this class. And then we say in the same book, in the second part, we're talking about immunity. So immunity just means how we preventing or against this illness to protect it of self, protect it of our body. So that means we study and learning microorganism. The arm is we prevent illness and protected human being. So then we go to a video, got some generally introducing for microbiology and how it development.
What is microbiology? Micro means very small. They are too small to see with our eyes directly. Bio means living things. They are including humans, animals, plants, and certainly some micro-living things. Logi means study. We are learning and study how the micro-living things reproduce, growing, and infecting humans, especially, some of them may cause serious illness. Microbiology is a science that studies the laws of life activities and the biological characteristics of various microorganisms. Microbiology is one of the branches of biology. It is mainly including bacteria, viruses, fungi, and others. It is a basic law to study the morphological structure, growth, and reproduction, physiological metabolism, genetic variation, and classification evolution of various microorganisms at the cellular, or group level. Medical microbiology mainly focuses on the presence and growth of microorganisms in the human body. Medical microbiology mainly studies the biological characteristics of pathogenic microorganisms related to medicine, the mechanism of infection and pathogenicity, basic theories of immunology, diagnostic techniques, and preventive measures to achieve the purpose of controlling and eliminating infectious diseases and microbial-related immune diseases and protecting human health. Microbiology applies it to science in many fields, like medicine, industrial fermentation, medical hygiene, and bioengineering. Biological products are widely used in people's lives. Most microorganisms are beneficial or harmless to humans, animals, plants, and only a few can cause diseases to humans, animals, and plants, such as typhoid fever, dysentery, measles, and polio in humans. Pathogenic microorganisms are called pathogenic microorganisms and are the main object of medical microbiology research. The purpose of studying medical microbiology is to understand the biological characteristics and pathogenicity of pathogenic microorganisms, like bacteria, viruses, and fungi, and to understand the immune function of the human body to pathogenic microorganisms. The relationship between infection and immunity, and their loss. To understand the laboratory diagnosis methods and prevention of infectious diseases in principle. What is bacteriology? Bacteriology is a branch of microbiology. Bacteria is the main microorganism to cause human illness. So sometimes people use bacteriology as the same as microbiology. The development of microbiology started with the invention of the microscope. In 1665, the Englishman Robert Hooke invented the microscope. He once cut a thin piece of cork from the bark and placed it under the microscope he invented. He observed plant cells, dead, and felt that they were shaped like single rooms where priests lived, so he used the term cell and single rooms to name plant cells cellula. It was the first time in history that cells were successfully observed. In 1673, the Dutchman Antony van Leeuwenhoek used his own improved microscope to observe real cells, along with recording the morphology of various microorganisms. He first observed and described single-celled organisms through a handmade microscope, which he called animal culls at the time. In addition, he is also the earliest scientist to observe blood flow in muscle fibers, bacteria, spermatozoa, and microvessels. (1798) British J.D. Edward Jenner is vaccinated with vaccinia for the prevention of smallpox. Jenner called his method vaccination. Vacca means cow in Latin. He then published a book on vaccination methods in 1798, an inquiry into the causes and effects of the variolae vaccine, a disease known by the name of cowpox, and for the first time, the word virus is used in the book. Jenner recognized the possible end result of vaccination. He hopes that one day smallpox will disappear on the earth. His dream finally succeeded with global efforts and cooperation. In 1980, smallpox disease finally disappeared on the earth. 1864 French Pasteur, Louis Pasteur, invented Pasteurization, a sterilization method for short-time heat treatment at 60 to 65 degrees Celsius to kill harmful microorganisms, and applied it to various foods and beverages. 1867 The Englishman Joseph Lister was the first person to use phenol as a disinfectant for patient wounds, inventing a method to avoid infection during surgery. Since then, strict disinfection systems have been gradually established throughout the world. 1876 German Ferdinand Kohn, who started to study bacteria, was also the first person to try to classify various bacteria in a systematic way. Bacteria can be divided into different species according to their morphological and physiological characteristics. Bacterial spores are found, thereby promoting the development of disinfection technology. 1884 Danish Hans Christian Gram invented gram stain. Gram staining is a method used to identify bacteria. This staining method uses the bacteria on the cell wall. The biochemical properties are different, and the bacteria can be divided into two categories, namely gram-positive and gram-negative. Originally used to identify the relationship between pneumococcus and Klebsiella pneumonia, it was later promoted as one of the important characteristics to identify the type of bacteria. It is widely used in the clinical diagnosis and treatment of diseases caused by bacterial infections. 1925 Alexander Fleming discovered that the antibiotic penicillin was accidentally during the experiment. Penicillium can produce penicillin, and penicillin has the effect of killing other bacteria. This is the first time humans have antibiotics. After the 1940s, the research results of microbiology were used in medicine, industry, agriculture, and other aspects. In biological research, molecular biology, biochemistry, cytology, genetics, and other disciplines also often use microorganisms as research materials. In the area of preventive medicine, the World Health Organization announced on October 26, 1979, that smallpox had been eliminated worldwide. Welcome.
Okay, so still coming back to this page. From the radio, we know there's a long history, the microbiology development. And also we say sometimes people use like bacteriology. That's the word, bacterial. Yeah, bacterial and learning, we know already. Yeah, bacterial and learning already. So that's basically sometimes the words we use the same way. We're still talking about microorganisms, still talking about the mainly things cause the people illness is the bacteria. So that's basically this class we will talk about and why we will be learned.
Sorry, I can't hear anything. It's just my problem.
Oh, okay.
I can't hear as well.
Nothing show up.
I hear. I can hear now.
You can hear. Can
Yeah.
you see? Can
Yeah,
you see?
yeah, I can see.
Okay. Okay. Yeah. Yeah. If sometimes you couldn't see or you couldn't hear, just let me know. Yeah. Sometimes I still have some trouble to control the Zoom. Yeah. Look, everything look like so complicated here. Okay. Okay. Then we talking about the microorganism. Micro still the same meaning. Yeah, Michael still the same meaning. And the organism still means the living thing. So microorganism, we're still talking about like bacteria, and we're still talking about virus, and we're still talking about fungi. Or sometimes people use the pronunciation fungi. And we also have some others still calls the ionists. Yeah, still calls the ionists. That's we say protozoa. Yeah, protozoa. We're talking about what's the difference with bacteria or virus later. And sometimes we have some worm and parasites. That's a little bit bigger compared with microorganisms. But that's still commonly easily cause people getting sick or illness. So we still put them in the microorganism that cause people sick or illness category here. So we say microorganism, usually we're talking about some small living things. They can cause people infection kind of illness. So that's what we're talking about. And the five pretty commonly kind of microorganisms we put together here. Then let's compare. Yeah, then let's compare. So we say we separate 5 or 4 different categories by the first one is look like by its size, yeah, diameter, size. We say the microorganism, so tiny small, yeah, so tiny small. The size you can see is how small it is, yeah, how small it is. So that means usually we're talking about 1 millimeter equal thousands here. So you can thinking about how smaller it is, how smaller it is. In other words, all the number here you time 1,000. And then you can think about how bigger it is. Yeah. So that's basically we're talking about. It's so small. We only can see it with microscope. So firstly, we see virus is the smallest one. Yeah, virus is the smallest one. It's a so tiny number in there. So tiny number in there. Bacteria compare with virus. Yeah, compare with virus. Little bigger, but still small. Yeah, still small. Yeah. And then fungi. Yeah, fungi. Fungi generally say let it be more bigger compared with bacteria. But still small, still need time thousands, and then look like 3 to 5 millimeter for fungi. And there are another two categories here, still smaller, still smaller. So that's basically, firstly, we separate microorganisms by their size, diameter. Then we separate it, or category by the cell, When we talking about the cell, we talking about the human body cell before in our pharmacology class. We say normally cell, we will have cell memory. And we will have nuclear. And we will have some liquid between, we call cytoplasma. And certainly we have some small organisms inside of cell in the cytoplasma. That's basically structure of the cell. But varies. Totally no cell. They don't have like a nucleus, don't like the whole form of the cells. Bacteria have form of cell. Fungi have it. And another two also have it. So that's another difference. In other words, why varies so small? They didn't have the cell structures. They just, we talking about like they only have DNA or RNA. We say inside the cell, in the nucleus, we have axon. We will have DNA there. And in the cytoplasma, we will have RNA there, usually. They are together perform the function, like we're talking about cell division later. Normal cells still have division, and they can form the two exactly the same cell. But for the virus, they only have DNA or only have RNA. It's depending different kind of virus. So some virus only have DNA. Some virus, they only have RNA. So how they can reproduction? They must be go inside of living cells, use the living cells, RNA or DNA, or maybe other like nutritions, and then they produce it. They make a copy itself for virus. So that's totally different with bacteria or with fungi. So that's virus. For bacteria and fungi and other microorganisms, they will have both DNA and RNA. So that's another difference. So that's about the size of the microorganism. Then we can see, some look like some shape in here. We're talking about shape. We finish size already. Of the bacteria. We especially go to bacteria today. Microorganisms, including we say commonly virus and bacterial and fungi. We're talking about bacterial first. Yeah, we're talking about bacterial for 2 classes today and another time on Thursday. Then we're talking about virus for another 2 classes. So now we especially go to bacterial. So bacterial. We classified by its shape into 3 groups. So if the bacterial, you can see the shape here, like a round shape here. Yeah. Whatever, they connected each other or separate. Yeah, they have different form. Different bacteria, sometimes they living separate, sometimes they living 2 together, sometimes they living look like a chain, sometimes the living look like a book. So whatever is the basic shape is round shape. So these kind of bacteria with special name it. Last just means round shape. So picture here, that's coming from truly the bacteria under the microscope. Yeah, under the microscope. So that's the round shape kind of bacteria. Some other bacteria, they may have different kind of form, but basically, like they are pretty long, compared with diameter. They may have different shape. Yes, sometimes like if you see this picture here. So from this picture, you can see we're talking about the size here. That's how long of the size in here and compare the bacteria. So you can see this one pretty similar, or even this one pretty similar like the shape here. But truly they may have some other shape, like a half moon kind of shape or some other shape. But whatever, we just put these kind of shape bacteria another category. And we also have some bacteria. Like kind of stand there. So like a picture here, yeah, all the picture here is the truly bacterial picture. And the picture here is the model. But truly you can see here that's the truly bacterial here. Yeah, pretty similar all pictures. Certainly they have lots of here. That's still under the microscope. Yeah, still under the microscope. We just gave it a name. So the words here is pretty close to Latin words. So that could be different with English words. But just the meaning and shape. So that's the bacteria. Basically, we say by the shape, we separate with 3 groups. Later on, we will talking about by the dye, we can separate another 2 big groups. But right now, we're just talking about by the shape, how we can separate. Then we're talking about the structure of the bacteria. Like what the bacteria look like. So certainly, this is a model. Yeah, this is a model picture. if we try to see like this kind of clear structure inside of bacteria we talking about before, we must use microscope. Yeah. Use microscope usually at least time 1,000 or even bigger, more than 1,000. Yeah, sometimes they use electrical microscope. It can go to 3,000 to 4,000. And then we can see the bacteria. So we compare the bacteria with normal cell. Yeah, normal cell. We're talking about the normal body cell. We say we have a membrane. We have membrane, and we have nucleus, and we have cytoplasma. That's like the liquid part. Yeah, like the liquid part in here, cytoplasma. And we have small organisms, yeah, in the cytoplasma. So that's the generally normal cell or normal body cell. So then let's compare here. Let's compare here. So the cell, bacterial cell, they still have membrane. Similar like the human body cell, plasma membrane. Yeah, plasma membrane. So that's this length here. And they still have kind of nuclear in there. Yeah, still kind of nuclear in there. Inside the cell still have kind of nuclear in there. And have DNA in there. But their nuclear is much simple compared with the human's nuclear. But still pretty similar there. Still pretty similar there. That's second part. And the third part still pretty similar. Cytoplasma. Cytoplasma. That's the liquid part here. Yeah, liquid part. Pretty similar like a human being cells. And they still have some tiny kind of small organisms. Yeah, they have simple compared with human beings. It's much simple, but they still have. So that's basically part will compare with the human body's cell. But for the bacteria, something will be different. Yeah, something will be different. What's the difference? Yeah, what's different? You can see on the outside of the plasma membrane. Yeah. On the outside of plasma membrane, they have another length here. Yeah, another length here. It's called cell wall. It's not in the human being cell. They have another protection or cover on the outside of plasma membrane. And it also, for some bacteria, yeah, not, so basically cell wall in most bacteria, but also some other special bacteria on the outside of cell wall, they have another cover there we call capsule. Not in all the bacteria. Some special bacteria, they will have another capsule. And the capsule then is much thinner compared with cell wall and compared with the plasma membrane. So like we use medication kind of capsule in there. And also for some bacteria, yeah, for some bacteria. They have one or even more, look like a little bit longer tail here. So we say not for all bacteria, but some bacteria. And also they have small like a body hair in here. They will all over the bacteria. That's still for most bacteria. We call Pilis. So that's the basically structure of the bacteria. Certainly, different bacteria, they may little bit different, but that's for general structure of the bacteria. Let's still looking for a video.
Let's talking about the shape and structure of the bacteria. It is important for us to understand how it causes illness. Bacteria are classified by shape into 3 basic groups. The first group, if they look round, we call them cocci. The second group, if they are rods, and we call them bacilli. The third group, if they are spiral-shaped, we call them spirochetes. In each of the category, they still show different form with microscope. In the cocci, some of them always gather together, look like grape. Usually we call them Staphylococcus. Staphylococcus. Look like grape. Some of the cocci may link together like chain, we call them streptococcosome. Streptococcosome. They're like chain. Some of the cocci are double together, we call them diplococcus. In bacilli, they still have different shape, some rectangle, some fusiform, some half-moon, some branching. The different shape we use to name the different bacteria. In spirochetes, they also show different spiral states. Now, let's talk about the structure of the bacteria. Different bacteria, their structure will be different. Like some bacteria will have capsule on the outside of the cell wall, but most bacteria don't have. The capsule is a gelatinous layer covering the entire bacterium. It may play a role in the adherence of bacteria to human tissues. Inside of capsule is cell wall. The cell wall is located external to the cytoplasmic membrane, provides structural support and maintains the characteristic shape of the cell. Inside of cell wall, it is plasma membrane. This is composed of a phospholipid bilayer. It performs the important functions: active transport of molecules into the cell and secretion of enzymes and toxins. The cytoplasm has 2 distinct areas when seen in the electron microscope. 1. An inner, nucleoid region composed of DNA. 2. An amorphous matrix that contains plasmids, which is a DNA molecule outside the nucleus, which has the ability to replicate autonomously. Ribosome is mainly composed of RNA and protein. In some bacteria, on the outside of cell wall or capsule, they have long whip-like appendages. It is called flagella that move the bacteria toward nutrients and other attractants. Some bacteria may only have 1 or 2 flagella, but other bacteria may even have more hundred of them. Bacteria also have hundreds of short hair-like filaments that extend from the cell surface. It is called pili. The main function of the pili is adhesion, which can make the bacteria adhere tightly to the surface of various solid substances. A specialized kind of Pilis, the sex Pilis, forms the attachment between the male, donor, and the female, recipient, bacteria during conjugation. This is the basic shape and structure of the bacteria.
So that's the basically structure of the bacteria. Then we will talking about some detail, one by one, by bacteria. So we say bacteria have a cell wall on the outside of a plasma membrane. That's for most bacteria, And inside the membrane, we have cytoplasm. And some tiny small organ in the cytoplasm. Cytoplasm. We're just talking about 3 of them, but truly more than 3. But we're talking about 3 mainly things. One is pretty similar like people's nucleus, but it's more simple. So we call it a nuclide. It's a little bit different. It's more simple. We're just thinking about simple nucleus. Then we have another 2 important, Robertson and another one. They still perform pretty important functions in the bacteria. We talk about some detail later. Then we talk about some structures on the outside of the cell wall. So we say most bacteria, they will have cell wall. And then they will have plasma membrane. Then they will have whole place here, cytoplasma. Cytoplasma is the liquid. And in the liquid, inside the liquid, we have look like a nucleus. Basically, it's a little bit bigger compared with the others. And certainly we have some others. Some little bit bigger and some little bit smaller. All over the body, cytoplasma. So that's basically for most bacteria. And then we say outside of the cell wall. So this layer of cell wall. So outside cell wall in here, we have capsule. So that's the first one. Capsule still the whole cover there. And then we talk about we have some tail look like maybe 1, maybe 2, maybe even hundreds. Let it be longer. This size let it be longer. And certainly they will have some pretty shorter one. Still all over the cell body. Still hundreds. Still hundreds. So that's the smaller one here. So that's the basically like most bacteria. Most bacteria. Like this. Like this. Certainly some bacteria will have all the structure. Some bacteria maybe have some of them. But for some special kind of bacteria, they have ability or function to form the spores. What means spores? Means they can change from bacterial to the spores, basically smaller and pretty thin cover, and they only keep maybe DNA in there. Too small here. Inside was spores, inside spores. And when the environment is suitable for their living, they can change back to the bacteria. During the spores time, they just stay there like stop metabolism. They holding there, but they can live in pretty long time. When they changing back to bacteria, they still can make a copy or division to more bacteria later. So that's the special kind of form of bacteria. They only happens in some special kind of bacteria, not for all the bacteria. So that's the general picture structure of the bacteria.